Okay, I'm going to talk about raster image correlation spectroscopy, and I'm going to tell you the story of how this came about. So I was a postdoc working with Enrico. I actually came down to his lab. I was working in Chicago, and his lab was in Urbana-Champaign, Illinois. So that's about 2 hours drive from Chicago to Urbana, in the middle of nowhere. So Illinois has a great city, and then the rest of the country, or the rest of the state, is like cornfields, right? So Urbana-Champaign is a great college campus, but there's nothing else around there. And so I was talking to Enrico about FCS. So I was doing these FCS experiments, and I like this quote by Erwin Schrödinger from 1943. How can the events in space and time which take place within the spatial boundaries of living organisms be accounted for by physics and chemistry? Right. So I was doing these single point FCS, and I was watching my cells. I couldn't see the cells while I was doing the FCS trace. And I was like, I want to see what's happening in the cell, right? And that was really important. And so FCS is a great method. It was developed by Elliot Elson in the 1970s to measure kinetics of molecular interactions. Then in 1995, in Enrico's lab, Keith Berlin, who was a student in his group, developed scanning FCS, which was great because instead of doing one spot, they figured that if they do an orbit of many, many spots, you can multiplex the spatial locations of your FCS trace and get much more information from one orbit. And that was great. Then Paul Wiseman and Niels Petersen, and you're going to hear a talk about one of Paul Wiseman's postdocs who's in the audience, Rodrigo, in 1995, published the image correlation spectroscopy method. And I remember when Paul Wiseman was giving his talk, he said, I recall Enrico coming up to my poster when he was looking at this correlation spectroscopy and saying, but Paul, this is very nice, but have you considered the time information of your data? And Paul was like, what is he talking about? Time information for my data. So Paul and Niels Petersen developed this way of correlating the intensity across the image so that you can obtain the size of molecules, how big the size of molecules are. And I'll tell you a secret. If you want to measure the point spread function of your system, one of the ways to do it is to get a molecule that's much smaller than the point spread function, like 100 nanometer beads, 200 nanometer beads, something that's small. And you simply fix it on the slide. It's not moving. And you take an image of that bead. And if you perform image correlation spectroscopy, or ICS, across that image of the bead, you're simply going to get the point spread function back. The diffusion will be 0, but you can actually calculate how big that molecule is. If it's bigger than the point spread function, you can actually calculate the size of the molecule, but because it's smaller than the point spread function, you'll just get the diffraction limit of your system, okay? That's ICS. And so Paul said, that's interesting, time information. So guess what? He went on to develop the temporal image correlation spectroscopy. This is also another part of the thesis that went into Claire Brown's research in Niels' group. And TICS then developed into STIX. And you're going to hear about spatial temporal image correlation spectroscopy, and that enables us to look at molecular flows, right? And then another calculation called the K-space or inverse space ICS that enables you to accurately transport measurements independent of the fluorophore photophysics. Could be a problem in many systems. All right, so there are a lot of drawbacks with FCS. I love FCS, but you're only looking at one spot unless you're doing an orbit or raster scan and doing a line as a function of time. You don't know what the cell is doing during that measurement. you can only look at one spot at a time. So for me is like this, right? For me is, I am looking at, oops, I am, oh no, what happened? So single point FCS, I just see one spot, and essentially in this spot, I cannot see what's happening in the field. So in the field, there are many players. You're all maybe soccer fans. So you might understand this picture where there's a lot of different things, a lot of molecules that are moving in different points. We have boundaries, we have edges, we have a goal, we know exactly that target for the ball to go, but we cannot see that in FCS. So how can we compensate for this? And how can we get information from images? And Enrico, then when I was working in his lab, he said, okay, Michelle, we have this wonderful 2-photon raster scanning microscope. And I think that we can get information from an image. And I was looking like the same thing as Paul, but the image is just an static image. It's just the intensity of the molecule there. And he's like, no. Never worry in advance. Just trust me. Take the image, and then we're gonna do this. And it's almost like he already knew exactly how the correlation functions were gonna come out from the image. So I'm gonna try to explain this to you. And at the time, this was in 2003 when I joined this group, 2004. And there were no commercial microscopes that can do FCS, right? You had to build this gigantic system to do FCS. And there are a few commercial microscopes out there. And one of our collaborators, Rick Horowitz, who was had a postdoc, her name was Claire Brown. We were, you know, two postdocs. I was talking to her, and I said, okay, Enrico wants you to do an experiment. I'll do an experiment on my side. You do one on her side, and we're going to raster scan. She's going to do the experiment on mine, and I'm going to show you the data from her system. So now you can actually do correlation spectroscopy from commercial laser scanning microscopes. It can be done whether it's analog or photon counting detectors, and it provides information. We can remove a lot of features from the image. We don't want to correlate the edges of the cell. We don't want to correlate, you know, the cell nucleus. We want to correlate just the fluctuations of the molecules. We have to do some immobile subtraction here, and we can provide information diffusion, not just diffusion, but we can distinguish diffusion from binding. So how does it work? Now, there's a very nice slide that Paul shared with me. When we're imaging, we're moving, as Lionel mentioned, we're moving the beam across the image and obtaining intensities. But this is what Enrico knew from their image, is that as we're scanning, the molecules are freely diffusing, right? So from one image, we're actually obtaining fluctuation of that motion of the molecule, right? And so it's just one frame here, right? So, and then the laser goes to the next frame, and it'll do the same thing. But what's exciting is that the fluctuations that you saw in this frame are captured in your intensity image. All right. So what we have is the laser scans across the line. It will retrace, we'll do the next line, and so forth, all the way till it gets to the end of the frame. But what's embedded in this is the time that we spend in every single pixel, right? And we know the line time, and we know the pixel dwell time from our image. So the actual time information is captured in that one image. So similar to FCS, imagine that we have these pixels now, and these pixels are smaller than our point spread function, our volume. So we can fit many pixels in our volume. And by doing this, all we have to do is reduce their pixel size. So if we go down to a pixel size of 50 nanometers, that's way smaller than the point spread function, which is approximately 300 nanometers, right? So now if we have a pixel 1 and the molecule is diffusing, what will happen is that we'll a high probability of observing that same molecule in pixel 2, pixel 3, pixel 4. If we scan slow enough and maintain the molecule fluorescence within our point spread function, right, we're going to have a high probability of seeing in this pixel, and this pixel, and this pixel, and this pixel. So we correlate the intensity fluctuation as a function of pixels. The higher probability of seeing the molecule, we see very good correlation, very close pixels, right? These are pixels that are very, very close together. And what happens with fast moving molecules is that if we're scanning very fast and the molecule is moving very quickly, we have a high probability of catching up with that molecule. So the correlation across pixels, adjacent pixels from this pixel to this pixel will narrow, but we're going to see correlation further in pixels. So you're going to narrowing of the function, but broadening as a function of pixel here. And this information, we can actually capture how fast the molecule is moving. And how do we do this in an image? The image has many pixels, right? So it's not just a few pixels. So we take the image here and we apply the correlation between adjacent frames. So we say, what's the intensity here correlating with the intensity here? Then we go to the next pixels and we keep doing all the way across the image. Then we do by 1 pixel delay between this pixel and that pixel, this pixel and that pixel. We also do the same thing in the y-axis. So we correlate in the x direction and in the y direction. And this is how it works. You only need 1 frame. The reason to take many frames is for statistics, but 1 frame, you have captured the diffusion. So in image number 1, we'll put let's say in matrix 1, and the same image number 1 in matrix 2. And so the calculation now is that in the shift in the x and y direction, if we shift so first, there's no shift, it's just the calculation of itself. So it's a 0, 0 point. It's very, very highly correlated. The amplitude's high. If we shift now over 1 pixel and we now 1 number is obtained for each pixel delay normalized by the average intensity squared, we normalize it to the average intensity squared, we get now the correlation shifted by 1 point. So we're starting to build our correlation function. Pixel number 2, pixel delayed by 3, pixel delayed by 4, pixel delayed by 5. And the further we go in pixels, the less probability we have seen that molecule. So we're building the correlation function along the x-axis. In the next line, we're going to do the same correlation between this pixel and that pixel. And guess what? If the molecule is moving really fast, we will not see it in the next line. So there will be no correlation along the y-axis. If the molecule is big and it's diffusing slowly, we have a high probability of seeing in the y-axis. So we're going to also get a correlation along the y-axis, right? So of course, we take many frames to get good statistics. And the function is this. If we want to, for those people who are looking at the math, if we consider the pixel sequence, it's contiguous in space, meaning that along pixels, it's contiguous. The time, though, we have to retrace to get to the next line, it's not continuous in time. So we look at the intensity at pixel 1, we multiply that pixel XY will be 1, 1 or 1, 1, 0 or whatever it is. And then we multiply. So it's the product of the intensity in the X direction shifted by 1 pixel. That's the Z here. And then the Y direction shifted by 1 pixel in the Z axis. That's the psi here. Divided by the average intensity squared, and that's normalized. So we can calculate along the X, along the Y axis. And of course, the probability kernel tells us that if we're looking at if the particle is a function of time, there are 2 parts, the temporal term and the spatial Gaussian term, right? This one is the amplitude is going to go down very quickly and broaden. whereas the amplitude here will go down slowly and then eventually broaden, right? So for any diffusion value, the amplitude decreases as a function of time, and the width of the Gaussian increases as a function of time. We also have to take into account the scanning parameter of our system, right? So this is the scanning parameter, independent of whether the molecules are moving or not, right? So we have to take into account the S factor. And here we have, if delta R is the pixel size, delta R, we need to know how big that pixel is because we need to be within the volume of the excitation. And then, of course, this just explains the terms. This is the calculation. Recall the amplitude of the correlation function from FCS gives us the inverse number of molecules. The same thing is true for RICS. So if we put here 0, G of delta of D and Xi is proportional to gamma divided by N. So we can obtain the number of molecules from this. All right, let's do some simulations. Here are molecules. This is what you would see if you could see molecules diffusing on a membrane. Molecules are slow. They have a diffusion of 0.1 micrometer squared per second. And you can see them very round, right? It's very round. So if we correlate across the X and a Y axis, the function will look like this because basically this is symmetrical. So if we cut this into a quarter, right, we'll see the function. This is a three-dimensional image. If we plot it in 3D, the high red value would be high amplitude as a function of X, and then also in the Y. So you have good correlation along the X-axis, good correlation along the Y-axis, and that's what the correlation function looks like, very round because it's slow. Now, the molecules here, these are 40 nanometer beads, and we're catching up with the beads, and they're still big enough for us to trace to the next line. So the correlation now starts to shrink along the Y-axis. We can fit this and we get 5 micrometers squared per second. Now, if you can see GFP, the green fluorescent protein, this is what it would look like. And it moves very fast, really, really quickly. As a matter of fact, so fast you'd never see it in the next line. So now the correlation looks like this, only along the X-axis, but not along the Y. And we get a correlation that gives us 90 micrometers squared per second for GFP in solution. All right. So just to give you an idea of what the molecules would look like if you could see them. Of course, you can look at the horizontal, the X correlation, and then the Y correlation. And this was very nice data that was made by Claire. And essentially, this is talking about 300 frames and 128 by 128 pixels. This is the pixel dwell time. This is important, how fast we scan, and the pixel size. Notice the pixel size is very, very small. It's 30 nanometers here. And we have kind of a rule of thumb. If you want to look at diffusions that are very fast, you should scan with the fast time speed. If you have molecules that are slower, let's say between 1 and 100 micrometers, cytosolic GFP or cytosolic proteins, you would scan a little bit slower. And if you have much slower diffusion molecules, let's say on the membrane, you should scan at a slower scan speed. The point spread function should be 3 to 4 times smaller than the PSF, which is approximately 300 nanometers. And the same conditions as FCS is that you cannot have too many molecules in the observation volume, right? So this is the point. If there are few of us in this room, if there's only 2 people in the room and one leaves, we could tell, right? But if there's 100 people in this room and one person left, very hard to tell. So the same thing, molecular concentration should be low. How low are we talking about? Actually, we could measure up to micromolar, and Rico would argue that you can probably do more with RICS, but also, you don't want to scan too slow because you'll get a function that looks kind of weird. You are not catching up with the very fast diffusion molecule. And you don't want to, let's say, separate the PSF the pixel size has to be too much. So if the pixel size is 100 or even 200, that's too big. You will not see good correlation. It's not enough sampling size in your volume. All right, so back to our Claire Brown story. So Claire was measuring GFP, and so Enrico says, okay, give me a image of GFP in solution. She said, what? Why do you want an image of GFP in solution? You will see nothing. And he's like, no, trust me. Take an image of GFP in solution and give me the data. So she did this experiment where she just took GFP in solution. And in her, back then, this was the state of the art confocal microscope. It was an Olympus FlowView 300. And I know a lot of you have different types of instruments, but this is like the very first beta version of confocal microscopes. And this was in 2005. And I was doing 2 photons, she was doing 1 photon. And we were more interested in her data because it was the first time we can do FCS in a commercial microscope. So she took this data and she's like, I see nothing. Enrico says, no, I see something. So I said, okay. We did the FCS, the RICS now correlation. You could see we were super excited to see this correlation function. We fit this to a spatial correlation function, and we got 100 plus or minus 10 micrometers squared per second. We're like, yes, we got actually pretty good data for free GFP in solution on a commercial microscope, first time ever. So, you know, Claire was very much interested in observing like how many frames, how many pixels do we need? Can we obtain the proper diffusion based on number of frames analyzed? So this is what this data is showing you. We have 256 by 256 pixels. We get a good G0 back, right? So no, not too much difference. And this is number of frames. So whether you do 1 frame or 500 frames, 256 by 256 pixels, and I think the pixel size here is 30 nanometers, you get good G0 values. But you start to get some errors if you take not enough frames. So you need good statistics. Make sure you take enough frames to get good statistical value. But then something happens because we're looking at GFP in solution. If your pixel size is too small, you can't correlate it long enough to see the diffusion. So the G0 is bad and your diffusion is really bad. Okay, so this is getting the diffusion back, number of frames analyzed, number of pixels. And I'm going to share this with you so you see those values. Can we get the proper concentration from the amplitude of the function? And if you measure GFP, and I said this experiment earlier, we'll take GFP, we'll measure it with the UV vis spectrophotometer, measure the absorbance, and calculate it in nanomolar. What do we get with RICS? It's almost a linear 1-to-1. The R squared here is 0.99. So if we get like, in this case would be 50 nanomolar, the calculation for RICS, the amplitude of the function is about 50 nanomolar. So you get almost 1-to-1 calculation. It starts to deviate lower concentrations for some reason, but it's still pretty good. Everything I've showed you so far is solution. How do we do this in a cell where we have a lot of problems? We have a lot of structures. In this cell, this is a cell expressing paxillin, and it has these focal adhesions. And it also has a nucleus, you see the edge, you see a lot of these features. We don't want to correlate this. So when we started this RICS analysis, we had to take into account detector noise. And that also gave us information of how to get rid of these immobile features, the features of the cell we don't want there. So let me explain how this is. In this case, if we have a photon counting detector, most of those detectors are noiseless in the sense that the shot noise does not correlate in space, right? So if we turn off our laser, and we just measure dark counts and we correlate in X, we get no correlation. But analog systems like Zeiss has an analog system, Olympus, Leica, all of them have analog detectors. And they do this on purpose because they don't want you to fry the detectors. So you have these analog detectors. And these analog detectors, some of them correlate in space. If you turn off the laser, you measure dark counts, it will correlate, and that's due to the shot noise. So we have to take into account that. And so what will happen is we'll say, okay, what's our dark correlation? Oh, it's about 4 pixels. So when we do our analysis from here on out, we'll start doing the fit at pixel 4. Like we will delete these 4 first correlations because we know that's noise. That's all that happens, and that's what this is telling us here. So how do we go from those cell structures? All right, so remember I said Paul Wiseman was doing ICS, image correlation? If we calculate raster correlation, even with time, it doesn't matter. And you have these immobile features. This is the cell edge here, and this is the nucleus. You're going to get the spatial correlation of the cell image. We don't want this, right? Maybe it's good for like other things like what Paul was doing. But because we want the fluctuations of the molecules, we need to subtract the average of our stack. And by subtracting the average, I'm getting rid of the static structures. So the nucleus, the things that do not move. And this is how it's calculated. So the average intensity of each pixel over a stack, and the intensity of each pixel minus the average intensity from the entire stack of each pixel gives us this. So we take the image number 1, we subtract the intensity of image number 1 minus the average, but we get negative values. So now we have to add a scalar average is added. And then when we add the scalar average, we get positive uncorrelated numbers. And what's left over is the fluctuations of our proteins, which is giving us here. So if the cell moves, we have to do something. Instead of subtracting the average stack, we need to do something called using the moving average. So in this case, instead of starting from image number 1, we'll start in image number 5. We'll average frames 1 through 10. We'll take image number 5, subtract the average of 1 through 10. Whatever's left will be our matrix, right? Remember the matrix I showed you earlier? That will be correlated in X and Y. Then we'll go to frame number 6, and I have it here, right? So this is frame number 5. This is the average intensity from frames 1 through 10. That's in our second matrix. Subtract that, add a scalar average, and this is what's left. Now, from here, we correlate just this in X and Y. But it's not enough because we have many more frames. So we'll go to frame number 6. We'll average frames 2 to 10, moving the 10 frame average. And we'll get another one that looks like this. Then we do the same for frame number 7. We average 3 to 12 and do the same thing. And we do that for all 100 frames. And by doing that, we can actually subtract out not just the structures, but the moving average. So here's a cell expressing a protein called paxillin. This is the structure. If I zoom in, I am able to get very small pixel size here. I think this is 50 nanometers. If I have no subtraction, I correlate this, I'm just going to get the Fourier transform of this structure here. But now I subtract the moving average. This is what's left. And I correlate this as a function of X and Y. And we get a spatial autocorrelation function after removing the moving average. We fit that and we get 2 diffusion values. 7 micrometers squared per second, which is our cytosolic protein moving around. And the protein that's bound to these focal adhesions. They're not membrane proteins, they're focal adhesion proteins, so it's very slow, 0.54 micrometers squared per second. And this was important to understand the dynamics of proteins that form these focal contacts on the cell. This is just to show you this. Well, we were interested to understand how paxillin formed the focal adhesions and the diffusion of paxillin across the cell as it's moving towards the leading edge. So here's a movie to show you this 256 by 256, 30 microseconds per pixel. And this was about a 5-minute movie. You see the cell here is moving towards this other cell. There's cytosolic proteins moving here, and you have many different focal contacts. So now when we analyze the diffusion in the cytosol, we get 10 micrometers squared per second. So this area, this area, this area, and this area gives us an average of 10 micrometers squared per second. These other focal contacts diffuse slower, 1.09 micrometers squared per second. And these guys over here have a different dynamics. They are even slower at 0.2 micrometers squared per second. So what we're seeing is that across this image, we can also look at the diffusion, we can map the diffusion value, which is here, how fast the molecules are moving, and the amplitude of those correlations. Remember I said that the amplitude gives us n, the number of molecules, but now when we subtract that moving average, that's no longer the case. We cannot backtrack and get that true molecular number of molecules. But I will, NMB, you can do that. And you will talk about NMB, right? Yeah. So in this case, we have the function here becomes the highest, let's say, probability of seeing molecular changes. So that's what the G0 is giving us. So this cell was retracting. Cells don't like the light, so they will go, move away. And these focal adhesions disassemble quite quickly. And so we know, and I don't have time to show you, but I'm happy to show you that how we calculated that molecules will add as monomers and then will disassemble as aggregates. And what happens is the cell is very clever. The cell says, I need to form a focal contact. It will not use all its resources to put one protein at a time. It will have to test that spot, saying, is this a good spot to attach? And if it's not a good spot, it won't attach. If it's a good spot because you have receptors there, it will attach. And then molecules can come in together and it's organized. When the cell needs to run away, move, it cannot do one protein at a time. It will take too long because there's too many molecules there. So there's a cleavage site, an enzyme that comes in, cuts it. And when it cuts it, these proteins start to fall apart. And they start to fall apart as large clusters, as aggregates. So we have this map. And now finally, can we do RICS on a smaller diffusion, like point spread functions? Of course, our lateral auto PSF is very small, but can we even go smaller? And of course, we can use stimulated emission depletion or STED. Now, if you have a STED microscope, you can also do RICS on it. The STED principle works by depleting, let's say, using some sort of faceplate to deplete the fluorescence, all but let's say the donut hole, right? So we don't deplete, so it increases the probability of exciting the molecule and bringing them back down to the ground state really quickly. So there's a dark ring, but only the center position is saved. So that becomes your observation volume, which is very, very small. Of course, the stimulated depletion works on this principle where a system's pumped by a second laser while the molecules are at the excited state, and it completely shuts down the fluorescence, right? The electrons go back down to the ground state, and then you have this stimulated emission. The spot size, right? Here's your excitation beam. You have a second laser that depletes the fluorescence, and the effective excitation is much smaller in the excitation, your diffraction limited spot, and it just goes down to very, very small. As you increase the STED beam, you increase the resolution here. And this is what a STED setup is. I'm not going to talk about that, but effective full width half max of your point spread function goes down as the STED power increases. So you get these beautiful super resolution images. And can we now do raster scan with our STED beam, and we can perform raster image correlation spectroscopy with STED. As you increase the STED power, we decrease the point spread function. And so the correlation, increasing the STED power goes much higher. All right, so this is now a cell. This was made by Nick Hede, who is one of our colleagues. And he showed that if you use ADO647 DPPE on the plasma membrane, this is on these XT cells here, without depleting the beam, you can still get correlation functions that look like this, but the volume is quite large and the correlation functions don't look very good, right? There's hardly any correlation here, for example. Using the STED beam, he's able to decrease the point spread function and get very good correlation in a much smaller spatial position, enabling you to obtain much richer information using STED imaging. Because now you have many places you can explore across the image, with much smaller diffraction limit. Okay, so overall, RICS enables us to look at diffusion using imaging. Certainly, you can do FCS if you take very fast images as a function of frame rate. So every single pixel becomes an FCS trace, which is really cool. And then you have to fit all that, right? So temporal ICM, if you don't use fast cameras, if you're using temporal raster scanning, you can still do correlations over time, but it's very, very slow in the order of seconds. RICS is much faster because it's within the same frame. So microseconds to milliseconds, and instead RICS the same thing, microseconds to milliseconds, but your spatial resolution is much, much higher. And you can use it to look at many proteins and so forth. And I'm gonna stop here, take questions, and thank everyone who was a part of this development and those who contributed slides to this presentation. And thank you for your attention.